my name is Albert van der Wal. I work with Elsevier. The part of Elsevier I work for is called Elsevier Life Sciences Solutions. And we help researchers in the life sciences area cope with the overflow of information. So we live in a world where every single day well over 5,000 new peer-reviewed articles are published in the biomedical domain alone. And our goal at Elsevier Life Science Solutions is to help researchers cope with this information. And we do that in different ways according to the different discipline we're talking about. So we've been publishing a database called EMBASE since the 1970s that does your traditional literature search, much like you would on PubMed, only it covers far more journals and it has deeper indexing. But in areas such as chemistry, our goal is actually to help researchers bypass the article altogether by giving them the data of interest at that point in time straight through the database rather than just serving them with a link to an article where they might find a boiling point or a melting point for that matter. In biology, we deploy a different strategy. We deploy our proprietary text mining technology. And there we go. What we do there is our technology can read papers. It reads millions of full text papers. It also reads the entire PubMed. And it can recognize not only entities or relationships between entities, but it can also characterize the relationship between two entities. So if you look at this sentence over here, Axin binds beta-catenin and inhibits GSK3 beta activity in hepatocytes. This single sentence contains two facts, and our technology can distinguish between these two facts, and it will distill, it will extract two relationships from that single sentence. And we use this technology to build a database by machine reading over 4 million full text papers, plus the entire corpus of PubMed. We've created a database of these types of relationships. And on top of that, we've created a visual modeling interface to help researchers with their work. And if we take a look at a typical use case, what we see here is an analysis done for finding out potential drug targets for melanoma. And the question being asked is, how do melanoma cells inhibit the local immune response? And this is a three-step approach. The first step is to find out from the corpus of literature which proteins are secreted by melanoma cells. And that information is in the database. So we have extracted those facts from literature. So you have the proteins that are excreted by melanoma cells. The next step is to figure out which cells in the human body are impacted by these proteins. And the third step is then to filter out only the cells that are inhibited by the secreted proteins that regulate potentially a melanoma cell. And you see that summarized in this visual, to put it very quickly. So the melanoma cell is central. Now each of these lines between the different entities represents a fact from our database, a fact that we extracted from literature. And now as a researcher, you might say, okay, I don't believe each and every paper we have. So you can hover over each of these lines and actually view the sentences from the papers that make that statement. And you can decide for yourself which of these papers you actually want to believe. Of course, they are linked to the original papers. You can filter out facts that are only supported maybe by fewer than 3 references. We have also an algorithm that allows you to filter out these facts based on a confidence level. And certainly do let us know when you come up with the reproducibility factor. then we'll be happy to try and integrate that as well into Pathway Studio. I mentioned we also have a database for traditional literature searching, which is called EMBASE. This is still done by manual keyword indexing, where we employ people to actually read full text papers and capture what the paper says. EMBASE is around because it supports a very wide range of use cases. So here in this particular example, I hope you can read it. It's just to figure out who is publishing about melanoma in Switzerland. And the Venn diagram, by the way, illustrates nicely why you should be including EMBASE in your literature search, because it tells you the number of references that are unique to EMBASE, and these are articles that you would not have found searching only in PubMed. So the keyword indexing looks like this when you look at a single reference. So for drugs and devices, we have particularly detailed indexing. We don't just capture, okay, which drugs are mentioned in a particular article or which adverse drug reactions are mentioned in that article. We actually capture what the article says about a particular drug from the adverse drug reaction point of view, and for devices as well. So this is important for a large segment of our customers in the pharma and devices industry that need to monitor not just their own drugs, but also competing drugs for safety reasons.